All right. Good evening, everybody. Happy Hanukkah. Be our last official new information class of the year, the semester, although we will have some reviews that we'll talk about in a second. Hope you're all doing okay. Hey, Alyssa. Hey, Maria. All right. Well, again, hope everybody's doing okay. So the exam is next week, final exam week. It will be available from Monday until I think like Monday first thing in the morning until Wednesday night. It's on Blackboard. It uses Honor Lock, but not Mastering Chemistry, just Blackboard. And I don't think it's quite live yet, but as soon as all the students get into it, there will be a whole new Blackboard shell just for the exam. So you'll get an actual new Blackboard class that says Chem 100 final exam that you'll be in. Hey, Joey. Hey, Daisy. So you can look for that to pop up. There's a practice exam in there as well. It doesn't have content to practice. It just is like to get used to Honor Lock and stuff. So just make sure everything's working for you if you haven't done that before. Some of the other sections, I think they didn't use Honor Lock at all this semester, so this is sort of brand new for some people. All right, so reviews. There will also be information of that when that course shell goes active, but I will just tell you, we'll have reviews Sunday at. Oh shoot! Now I forgot the time. Hold on. Sunday afternoon. I have the time here. Sunday at 4. Sorry, Sunday 3 to 4. Hey, Anna. The reviews will be Sunday from 3 to 4 right here, and Monday from 7 to 8. And those are going to be open. I mean, we're always public, but those will be open to all Chem 100 students. So there might be some more people around. If you want to meet with me at any point between now and then over WebEx or something, you're welcome to do that as well. Just let me know, and we'll find a time. All right, so hopefully all the studying is going okay. The final quiz should be up now in Mastering Chemistry. Although I think I just looked at it and it's not in Blackboard yet, so I'm going to have to put that over. But it is, if you actually go into the Mastering Chemistry section, it should be over there. And that covers the biomolecule stuff that we've been talking about for the last couple weeks. All right, so we don't have a whole lot more to talk about with DNA structure. Not that it's not important or not a lot of information, but for the purposes of our class, you saw the learning objectives are fairly fairly sparse. There's not a lot there. So let's just talk basically about some of the language here and some of the structure, and really, I think most importantly, look back at the rest of the stuff we've done this semester and talk about how these structures compare to the stuff that you already know. So what you see over here on the left is what we call a nucleotide with a T, nucleotide. And it is made up of 3 parts, 3 molecules that I'm not going to say look familiar, but that we can sort of tell apart. So we've got phosphate over here, which is a PO4 molecule with an OH here. Then we've got a deoxyribose sugar. Now, you've probably heard that deoxyribo term before because that's that first word in DNA. But now maybe you know a little bit about what that might refer to. So ribose is one of the sugars, one of the monosaccharides, and in its ring form, it looks something like this. And because it's deoxy, it's missing an oxygen. It's actually missing an OH. So if we had normal ribose, we would have what we call RNA, but we have deoxyribose, and then we have this thing called a base. So we know what bases are. In the context of DNA, the bases are these fairly large molecules that have multiple nitrogens on them, and there are several different ones. And all 3 of those things together are called nucleotides. Now, if you just have the sugar and the base and not the phosphate, Then you have a nucleoside with an S. So you can see that term here. So just watch that. That can sometimes be a little bit confusing. Nucleoside is just these two parts, and nucleotide is including the phosphate. All right. So the 4 bases in DNA are cytosine, thymine, adenine, and guanine, which look like this. They all have multiple nitrogens and some oxygens, some NHs as well. And as we'll see, that's really important for hydrogen bonding, something that we've definitely talked about quite a bit in the past. So the hydrogen bonding comes from these NH bonds on the bases and the other molecules that they can hydrogen bond to on those bases. All right. So getting back into the structure a little bit of how these things are put together. If you read through this, there's a lot of big words in here, but my hope is that after this semester, some of these big chemical words are not quite as completely unreasonably weird as they might have been in the past. So, for example, a phosphodiester bond. Certainly, that's a big word, but let's break it down, and you can probably figure out what's going on here. We've got phospho, so phosphorus, and then diester, so 2 esters. So let's take a look at that structure and see where that comes from. If we look at the way that these nucleotides come together, you can see that you have a phosphorus here with a double bond to an oxygen, and then another oxygen and another oxygen, and then beyond those oxygens are carbons. And so that's very similar to the ester functional group that we talked about with carbon. Ester functional group being carbon double bond oxygen, and then another oxygen, and then another carbon. So if we replace that with phosphorus, we have a couple extra oxygens there, but that idea is still the same. So there's our phosphodiester bond, one ester going up to one deoxyribose sugar, and the other ester going to the other deoxyribose sugar. And then so for each of these, they have one of these bases hanging off of them. And then we can skip this part about kind of the overall structure and where that comes from. It's interesting. You can read it, but it's not. Maybe we'll get to it in a minute. If we zoom out, this is kind of on the left here. This is your typical what you picture when you picture DNA, I assume, the double helix structure. But now that we know something about the chemistry, let's really zoom in and see what causes it to do that and what those different parts are. So if we zoom in on the double helix, we get something like this picture on the right. So let's break this down a little bit. So just looking at one side here, you've got a phosphate right there, and then that connected to the deoxyribose, and then to another phosphate and another deoxyribose, and so on. There's our phosphodiester bond that we just talked about that links those two sugars together. And then each of those sugars has its base. And then so here's what comes together then. The other chain is that same thing basically, but kind of pointing the other direction. So kind of like upside down. And these bases form complementary pairs. Let's see if I can get that a little bit bigger, based on hydrogen bonding. So you can see like up at this top one, you've got adenine and thymine that are hydrogen bonding with each other. So the hydrogens from the NH here can hydrogen bond to the oxygen and here to here, and they make this nice little connection. Same thing with cytosine and guanine that connect the hydrogen with the oxygen, the hydrogen with the nitrogen, and those dotted lines again are those intermolecular forces, those hydrogen bonding. And so only those two bases can bond to each other. So A goes with T and C goes with G. So sometimes called the base pairs, and that's what holds those. That's what holds that whole strand together. And then in that twisting structure, you have this major groove and minor groove where other stuff can happen. So proteins can go bind in there, or RNA can kind of get in there and start breaking this thing apart. So that's the basic structure for DNA. All right, and there's a close-up of those hydrogen bonding interactions between the base pairs. All right. And I don't think we... Yeah, so we don't even really get into RNA here in this book. But basically, the way that DNA can replicate itself for cell division is for this interaction to split and then instead have get transcribed into RNA, and then that goes on and does the cell division. As you can maybe guess here, I am a little bit out of my element here. There are many areas of chemistry that I would be happy to tell you about in great depth, but this is not one of them. So that's what we've got, and honestly, that's about as much as I know as well. I really need to work on that stuff. But that's all we are really concerned with for this class anyway. So that's all the new stuff for now. Do you have any questions about this? Or we can just jump right into any kind of review or other stuff that you want to talk about for the end of the semester. So I will watch the chat and wait for your questions. The last few labs should be graded soon. I have them mostly graded outside of Blackboard. I need to do an update to send all the scores up to Blackboard. So you should see everything, this weekend before the exam, ideally, unless there's a problem somewhere in there. Lots of polarity questions on the final. Yeah, I think. I don't know exactly what's on there, but it's a pretty good mix from the semester. Polarity comes up a lot. So like I'm not sure if you have a specific type of question, but the like there certainly are going to be questions about intermolecular forces. That's come up a lot in the biomolecules as well. So like looking at polar versus nonpolar, how that affects boiling point, how that affects solubility, why lipids are the way they are because of their nonpolarity. So a lot of that kind of stuff. Polarity kind of gets its way into a lot of other things. So, there may not be a lot of specific questions like, is this molecule polar or not? But I bet there's a lot of stuff that is related to polarity, I guess. So, for example, we talked about amino acids. We talked about polar versus nonpolar side chains. So something like that might come up. We talk about lipids. We talk about nonpolar, that kind of stuff. So if that helps. The nitrogenous bases. So should you memorize them? You should. I would say no. You don't. You certainly don't need to memorize the structures. So there won't be a question like, what is this base? Is it adenine, thymine, guanine, or cytosine? You should know that there are 4 bases in DNA, and their names are adenine, thymine, guanine, and cytosine. And you should be able to know kind of the idea of how the structure is put together. So like the hydrogen bonding interactions there. So more like recognizing in general. It's not an objective that you absolutely know like how to draw the structure, how to identify the structure of each different base. Though if you're going on and you have some biochemistry in your future, you might as well learn it now because you're going to have to know it then for sure. So I'm not sure where your programs take you in the future, but if there's a biochem class in there or a genetics class in there, something like that, physiology even maybe, you'll probably need to know that. Molecular biology. Skip a question and go back. Yes. So the exam will be like it will just list all the questions at once. So you can jump around however you want. It's not like mastering chemistry that only gives you one question at a time. There are no drawing questions. There's no way to draw in Blackboard. So there are you'll see when the shell pops up, there's an example of each type of question, but there's multiple choice, there's fill in the blank, and there's some calculation ones where you put in like a number with units. What formulas should we know? Well, let's take a look. Yeah, you're going to want to know molarity. Remember that this is all going to be based on the learning objectives. So if we go into Blackboard here and look through these bullet points that are in each section. So if you had a question like molarity, I believe that came up in the solutions chapter here. So you can see that you should be able to define molarity and calculate the molarity of a solution. You should be able to calculate mass percents of solutions and so on. Oh, it's up now? Okay, great. So this should really be your goal about what you need to be able to do. So for example, when we looked at unit conversions, the relationship between millimeters and centimeters, milliliters and cubic centimeters, those are given in the objectives. Like you have to know how to convert between millimeters and centimeters. Back here with number 1, you have to know how to deal with prefixes kilo, centi, milli, and micro. But for instance, there is no objective that says you need to know how many inches are in a centimeter. So you would not have to know how many inches are in a centimeter for the exam. So like hopefully that helps. You should be able to go through this, and most of these I bet you can do fine if you actually go through each of these lists with, or this whole thing, each of these bullet points with a pencil, and check off the ones that you know. I bet you know most of them without even studying, and then I bet there's probably a few in each section that make you go, yeah, I probably need to work on that. And there's maybe just a couple across the entire semester that makes you say, okay, I'm really gonna have to put some work into to make sure I can do this. Like for example, if at this point you still are not really comfortable with the naming things that you see here, the polyatomic ions, the naming and all that stuff, so now's your time to make sure you go back and review that stuff. All right, so answering questions. You won't be able to have notes out. No, you won't be able to have notes out. There's a periodic table on the exam. You are allowed to have a calculator, and you can have scrap paper to like write stuff on, blank paper. The number of questions, I'm not sure. I believe it's 50 something. 50 something questions. And the test will be graded based on the highest score. Sort of. So the test will be graded based on the overall statistics. So we will look at the mean, the median, the standard deviation, the minimum, the maximum, and we will adjust everybody's scores based on that. So it's not as simple as just like, oh, whatever the highest score is, we give everybody that many points. We're going to go back and really do a full statistical analysis of it to figure out how it should be adjusted. The reason we can do that for that and not our other exams, 67 questions. Thank you. That was close. We can't do that for our normal exams because there just aren't enough people. So the statistics aren't valid. Like we could just have a really, I've had classes where like the average is super high except for a couple people. And it's just because of who happens to be in that class. So when you have a whole bunch of people across all the classes, then the statistics make sense. Then things like the average are useful in figuring out how you should scale the test. So that's why we do that. Oh, good. So you got the announcement now. That's good to know. else? Other stuff you want to talk about? The exam, there's no oral option for the final. It's standardized for everybody. The exam is due it should say in that announcement. I believe it's Wednesday night at midnight, but let me check that to make sure here. Oh, it's not midnight. No, it's 10 o'clock p.m. 10 p.m. Wednesday night. It's due. No, there's no option to answer and then correct it later for partial credit, like in the second one. Oh, I think that was the first one where it took away some points, but allowed you another chance. This is just a standard, straight ahead, go through the exam and do it. It's 2.5 hours. So here, I found the announcement here. For those of you who haven't seen it yet, you should be in there. So definitely check if you have access to this shell, this new shell that's called Chem 100 Fall 2020 Final Exam. And so you have all the details here. It'll be available 12 a.m. Monday morning until Wednesday 10 p.m. And once you open it, you have 2.5 hours to complete. And I'm sorry, handheld calculators, oh yeah, are allowed, and HonorLock provides a scientific calculator if you need. Exam 2 answers, I thought I posted them. Can you not access them? Let me jump back over there and see if I can find them. No, you're right. It's not there. I mean, not that I was doubting you, but I thought I had it there. All right. I'm not sure what happened. I will. I'll try to get that up for you. Keep bothering me about it if you don't see it. And yeah, I'll make sure to get the oral exam ones up too. All right. Yeah. Sorry about that. I'll get them up for you. Other things for tonight. My goal is to have everything done before the exam, before you start the final, so you know kind of where you're at before the final. And then all we have to do is put in the scaled final score once that's done on Wednesday, and then everything is done. So that's the goal. Hopefully I'll be able to do it unless something happens this weekend I can't get to it. But that I want you to be able to see your grades before the final exam. The final 12%. I'm not sure. It's whatever it is on the syllabus. It hasn't changed. So let's see here. Yeah, 12%. Yep. The learning objective of stating the difference between a strong and weak acid or base. Yes, definitely. I'm just getting up some paper here. All right. So there's a couple things here. First is just the definition. What is the difference between strong and weak acids and bases? I think that's really what the objective is speaking to here. So a strong acid or base is one that dissociates completely in water. So that means it splits apart. So for example, HCl is a gas. When it dissolves in water, sorry, that's G for gas. When it dissolves in water, we have aqueous HCl, which is a totally different thing in that it is H plus and Cl minus separate. Or more correctly, it is hydronium ion because the H plus is gone with the H3O or the H2O to make H3O plus and chloride ion. But the point is that when you put HCl in water, there is no, remember, HCl is a molecule with a covalent bond like this. There's none of this left once this goes in water. It's all become ions. So Alyssa's question is the second part of this. So the first is the definition. The strong acid or base dissociates completely in water. Weak acids and bases don't. So weak acids and bases are mostly stay as their molecule and partially dissociate, partially don't. So the key to determining that then is to recognize that almost all the acids that we will deal with are weak acids, except for the strong acids, of which there are only a few. So HCl is a strong acid, HBr is a strong acid, HI is a strong acid, nitric acid is a strong acid, sulfuric acid is a strong acid, and perchloric acid is a strong acid. I don't think we really talked about that one this semester. But these are the strong acids. Other acids like HF, like acetic acid, all the organic acids, amino acids, anything else that could potentially give up hydrogen is carbonic acid, like we talked about in the bloodstream to regulate pH. These are all weak acids. So anything else is a weak acid. Phosphoric acid, all that. So there's like these few strong acids, and everything else is a weak acid. Everything else only dissociates partially in water. Now, I also want to just gently correct Alyssa's question. I know you didn't mean to say it this way, but dissolve, remember, is different from dissociate. All of these things will also dissolve in water, meaning that they will be absorbed into the they'll be absorbed into the water, and water molecules will surround them, and they'll be in solution, but they won't have split up into ions. Right? So same thing like with when we talk about strong and weak or electrolytes. So something like glucose, sugar, or sucrose dissolves in water, but it doesn't break apart into ions. So it's not an electrolyte. And so it's the same thing with the acids in the others, the strong acids in the others, which I don't know why I said others. They're the weak acids. They dissolve in water, but they don't dissociate in water. They dissolve in water, but they don't break apart into ions in water. And that's the difference. The misconception there is that when we think of the word strong, we think of like the effects of the acid. So we think of strong acids as being more corrosive, but that's wrong. The strong and weak designation refers to the electrolytic activity. Is it a strong electrolyte or is it a weak electrolyte? All right, what else? If you don't have anything right now, that's fine also, because we'll be back Sunday afternoon and Monday evening for more questions. Sure. Everybody good for now? Ah, great. Okay. For identifying compounds in the objectives, it just says describe ionic bond and ionic compound. Um, yeah, so like what is an ionic bond? What is an ionic compound? It's just an attraction of a cation and an anion to each other. Um, and for the compound, you'd say it's in a particular ratio. So like CaCl2, you have 2 chloride ions for each calcium ion. And yeah, let's talk about a catalyst. So a catalyst is something that you add to the reaction that is not used up in the reaction, but changes the um, mechanism by which the reaction proceeds. So instead of like 2 molecules bumping into each other and reacting, maybe one molecule bumps into the catalyst and then the other molecule bumps into the catalyst, and that allows it to do some sort of reaction. And the advantage of that, or the main point of the catalyst, is that it reduces the activation energy of the reaction. So if you had a reaction that, let's say, had something like that's our reaction. Remember that what that means is the reactants start at this energy, and then they have to get a bunch more energy to kind of get over the hump. This is energy on the y-axis. So they have to get a bunch more energy. This amount of energy that they have to get just to start going, to kind of like bump into each other properly, that's called the activation energy. And so a catalyst lowers that activation energy. So instead of having this much, the catalyst allows it to go through a different mechanism and instead maybe do something like this. And lowering that activation energy, so now the activation energy is like that increases the reaction rate. So by adding that catalyst, you've reduced the activation energy and increased the rate. So the reaction is now faster. It takes less energy to activate it. Notice what doesn't change. The overall energy of the reactants and the overall energy of the products does not change. Excuse me. So the overall change in energy doesn't change. Only the activation energy changes. So then to answer Alyssa's question, endothermic and exothermic is a way of describing the overall energy change of the reaction. So in this particular reaction, the reactants are at this level and the products are at this level. And so the difference between those is the overall energy of the reaction. Since it goes from higher to lower, that means that energy is released, and that's exothermic. If it were endothermic, it would be the opposite, so it would look more like this. So our reactants are down here, our products are up here, so we have to actually add energy in between reactants and products to do this reaction. So exothermic, endothermic. In a reaction like a chemical equation, we can also express that by putting heat in the equation. So we can say something like A plus B equals C. That's our equation. If it's an exothermic reaction, then the heat will be a product. It'll come out. If it's an endothermic reaction, then the heat will be a reactant. So you have to put heat in to get that to go. One thing that can be a little bit confusing about that is this idea of activation energy. And the easiest way to think about it is just to keep them separate. They're not perfectly separate because of some things, but if we keep them separate, we won't get confused. So activation energy being high or low doesn't affect the exothermic or endothermicness of it. So whether this middle hump is really, really high or not doesn't change whether it's exothermic or endothermic. Doesn't change whether it's ultimately going to release energy or take in energy. So where this sometimes confuses people is, for example, if you think about like burning a fuel, combusting a fuel, that's very clearly exothermic because it gives, as the fuel burns, it gives off a whole lot of heat. But sometimes people say, well, it's endothermic because you have to light it. You have to put energy in to get it going. And it's true that you have to put energy in, but it's not endothermic. All you're doing is providing the activation energy with that first spark or whatever. And then the energy that's produced by that reaction is the heat byproduct, the heat product that would make us say that it's exothermic. So think about the activation and the rate of the reaction as completely a totally separate thing from the overall heat of the reaction. Is it overall endothermic or exothermic? Does that help? Okay, good. All right, what else can we talk about? State which of the following substances usually dissolves in water: ionic compounds, polar molecular compounds, non-polar molecular compounds. So here's where we're thinking about similar intermolecular forces. So generally, we would say that water is polar, and so it's going to be able to interact with other polar molecules as well as ions because those are charged. So ionic compounds and polar molecular compounds generally dissolve in water. Non-polar molecular compounds generally don't. Now, there are exceptions there. There are ionic compounds that are so tightly bound together that they don't end up dissolving in water, um, precipitates and stuff like that. And as we've seen, there's other kinds of exceptions and things too. But with that term usually or generally, we can say generally ionic and polar stuff is going to go together okay, and non-polar stuff is not going to mix in with that as much. Yeah, that's a good one. So, um, this is this idea, and I think there's a good picture in the book here too, that's linked under that section. But if we have, let's say, a chemical equation like this, we've got our reactants and our products. And let's say now we're going to think of this more as an equilibrium reaction. So it's a reaction that's going back and forth. All right, so any reaction like this is going to, let's say we just mixed A and B. If we mixed A and B at the very beginning of the reaction, then we're going to start forming C and D. And at some point, we're going to form like a little too much C and D, and it's going to go back and form some more A and B until, and it's going to keep doing that, keep going back and forth a little bit until it reaches that point that we call equilibrium, which doesn't mean exactly in the middle. It doesn't mean we're going to get half A and B and half C and D. It could be anywhere along that continuum, but at some point, it's going to stop changing. Right? It's going to like balance itself. And so the analogy we always use for this is like two buckets of water connected by a tube. And if you like, so let's say there's some level at which they're balanced. They don't have to be even, but there's some level that the water level is balanced. So then if you were to add more water in the left side here, you would expect it to get kind of pushed over to the right side until it rebalanced. The same idea, if you took water out of the right side, then you'd expect the water from the left side to push over to kind of compensate for it until, again, it's balanced again. And the same thing happens with a chemical reaction. If we add more reactant, more of A or B, to the system, then we're going to make more of C or D. If we add more C or D, then we push the system back the other direction and make more A and B. And then the same thing if we take stuff out. So if we take some of the C as it's formed and we pull it out of the reaction, then A and B can be kind of pushed to form more. So that's Chatelier's principle. This idea that you can control the equilibrium by either adding or taking away from either side. And it's not just reactants, by the way, it's also things like energy. So in our previous example, we were just talking about exothermic versus endothermic. So let's say you have an endothermic reaction like this, or sorry, an exothermic reaction like this. So if you remove the heat from here, then you can make this reaction go more to form more heat. Um, Sometimes we don't think about it that way because generally we're not doing a reaction in a perfectly closed system where the heat can't escape into anything. But if we were, then we would have to remove that heat to get that reaction to keep going. So it's not it I guess that's written slightly confusingly because it says use the principle to determine the direction a reaction will proceed. But it's more like if you have a reaction that's balanced, how is it going to change when you add or subtract parts from it? Yeah, that's the idea. All right. I think then we'll say that that's good for tonight. Um, Let me know what you need in the next few days. I'll make sure to get those exam keys up there. I don't know what happened there, but I'll get those for you. Uh, Hopefully, I'll see you at the reviews Sunday afternoon and Monday evening. If not, I hope the exam goes well for you. And yeah, let me know if you need anything. And hopefully, everybody does awesome. And I can just enter just all A's. It would be so much easier. Just get all the A's so I can just put them all in that way. All right, thanks. All right, well, thanks everybody. I guess this is it. So we should say goodbye for the semester. Thanks for a weird semester. Sorry it was weird, but it is what it is. We did our best, and hopefully, maybe someday we'll get to meet in person again on campus. So thanks everybody. Good night. Thank you.